We're going to talk about the lymphatic system a little bit in this lecture, mostly on the anatomy of it. Now, there's not a lot of anatomy that we do on this in lab. It's mostly focused on the histology because the models don't show the lymphatic system too well. So the main things from a gross anatomy model perspective to know are the location of the spleen, the thymus, and like general areas of lymph nodes that'll pop up in a second. And then there'll be the histology of the spleen, thymus, and lymph nodes that we'll need to look at in more depth. All right, it's just a overview of the lymphatic system. So there are two different parts of this. There's the plumbing side of it, the network of vessels that we'll look at in the next slide. And then there's also the tissues, the cellular component that we see kind of using or hijacking the lymphatic vessels to keep an eye on what's going on with the pathogens and cell debris and external, or internal, not external, internal environment of your body. So the main function of the system is to return those interstitial fluids, which are a large part leaked plasma from our capillary beds, back to the blood. You do get a little bit of proteins that do escape from those capillaries sometimes, not a ton, but those would accumulate in the tissues, and so the lymphatic system helps to return those too. It also plays a role in the immune surveillance side of things, and then our return of lipids back into the cardiovascular system as well. Normally, digestive products, carbohydrates, proteins, nucleic acids, they're collected in capillaries in the digestive system that go to the liver. However, with lipids, those diffuse into the lacteals. Those are lymphatic capillaries in the small intestine. And so lipids actually go through the lymphatic system and then are dumped into the blood in the subclavian veins and then get circulated around the body. So that is something that is specific I would like you to know. The fluid that is moving through the lymphatic vessels is known as lymph. It's essentially interstitial fluid, has much less protein than blood plasma does. Again, a little bit for whatever leaked out, but generally a lot less. But the ion concentration will be pretty similar. There'll be glucose, there'll also be waste products. So here we're seeing the lymphatic system. Again, a lot of this stuff is not named and not on our list because what we need to do is really just kind of get an idea of the main areas. Lymphatic structures can be a bit variable in where they pop up. So we focus more on like, what are the areas we find a bunch of lymph nodes, or what are the really big structures that are consistently present in the lymphatic system? And when we look at that, you can see the different regions. So the cervical, axillary, and inguinal are some key ones where we do find these clusters of lymph nodes. These tend to be places where a lot of lymphatic vessels come together from the appendages or from the head and neck area. And that makes just a nice place to set up watch points or lymph nodes in order to catch anything that might be moving through that. You also have quite a few that wrap around the aorta in the abdominal cavity, and those are important for picking up stuff that might have creeped through the digestive membranes. As far as main structures to know on this, so we do want to understand where our cisternae chyli is. This is the main collecting duct for those abdominal and lower appendage lymphatic vessels. We also want to then know the thoracic duct, which collects lymph from the left side of the body, so left head, left neck, left arm, left torso, and both legs. And then the right lymphatic duct, which only collects lymph from the right arm, right torso, really right kind of thoracic cavity area, right head, right neck. And so that is a key difference. Unlike a lot of other things that are, fully bilaterally symmetrical in our body, the lymphatic drainage is not. Looking at the microscopic structure of lymphatic vessels, these are structurally really similar to what we see in the venous circulation. Lymphatic capillaries look a lot like our normal capillaries, except they're a little bit leakier, so a little looser junctions between the cells to make it easier for interstitial fluid to move into them. They're also anchored into the surrounding tissue via filaments that help to open up clefts or gaps between the cells. And those lymphatic capillaries are blind-ended, unlike our systemic capillaries and pulmonary, where you have blood going in and out through a closed vessel. Lymphatic capillaries are just this kind of dead-end alley that's got leaky walls that then starts up and feeds that lymph into further lymphatic vessels. And it goes from lymphatic capillaries to lymphatic venules to lymphatic collecting vessels, which are like lymphatic veins, and then our trunks and ducts. Everything from the lymphatic venules up will have valves in it, like the venous system too, and that helps to make sure lymph moves only in one direction. All right, I think I mentioned most of this in the previous slide, so I'll just move on. Same thing with this, mentioned in the previous, well, 2 slides ago now. Here's a picture of our lymphatic capillaries, and what I do like about this is it shows those anchoring filaments that tie into the extracellular matrix of the interstitium. And those mean that when your tissue moves, it helps to open those little clefts between the lymphatic endothelial cells. And that just helps to facilitate return of lymphatic fluid. And is one of the reasons why like getting up, moving around, or getting massages can help increase the flow through your interstitial fluids. So like if you had a really big workout, maybe there's some lactic acid and pH changes that accumulated in the interstitium between your muscle cells. Well, you get a massage and it can liberate some of those or increase that lymphatic return from that area, which gets you fresh new fluid in and just can help kind of wash out stuff. So there is some good evidence to suggest that those sort of deep tissue massages can increase lymphatic drainage. That would also help with things like compression socks or flight suits. If you look at pilots that deal with, potential interstitial fluid accumulation. Okay, so during inflammation, those lymphatic capillaries can play a bigger role as well in that they're picking up cell debris and pathogens and cancerous cells, and then that lymph goes through these nodes where you get this nice examination or like checkpoint function of lymphatic tissues. Here the lacteals are mentioned, but we already talked about that. Our collecting vessels, they're histologically like veins. It's very difficult to tell them apart unless you see if there's blood in them or not, although they tend to have a little bit thinner walls than veins and a little bit more valves because the pressure inside the lymphatic system is so very low. Our lymphatic trunks are the big ones. They're formed by the union of the largest ducts, and so we have the 2 large trunks. And the terminology gets a little weird. I'm not going to mark you off if you put thoracic duct or thoracic trunk or right lymphatic duct or right lymphatic trunk. But these larger structures are in the trunk/duct classification. And this is just a closer view of the right lymphatic and the thoracic duct and where they drain into, which is kind of like the armpit, if you will, between the jugular vein and the subclavian vein. As far as lymph transport, it is essentially the same as venous return mechanisms. So you're going to have, it doesn't really pump, although the vessels do weakly have a peristaltic motion, so they weakly contract, and that can help return some of that lymphatic fluid. But pulses in nearby arteries, which I can't remember if we talked about in the lecture, but that's actually a subtle but interesting way that you can increase some venous return or lymphatic return. And then you'll also get contraction of muscles, and the thoracic/respiratory pump will affect lymphatic vessels in the thoracic cavity. So similar to the things that affect venous return, you'll see that with the lymphatic system. As far as our lymphoid cells, the main ones that we'll talk about are going to be the T cells and B cells of the lymphocyte lineage. We do have NK cells, but those ones aren't easy to identify, so for lab, we're not really going to spend a lot of time on them. T and B cells will protect the body against antigens, and hopefully those antigens are also pathogens, things that we don't want inside us. Sometimes they can misidentify your own cells, as antigens, and that's when you get autoimmune diseases developing. So again, hopefully those antigens are actually foreign, but sometimes not so much. This is also the basis for our blood typing test that we did in week 3. Our T cells, those ones are going to be a combination of helper cells, suppressor cells, and our cytotoxic T cells. Cytotoxic T cells are the ones that go out and they really do the direct hand-to-hand fighting with other cells. They're really good at telling our own cells, hey, you need to commit apoptosis because something's wrong, you're infected or cancerous or whatever. The helper cells are critical for activating the immune response both with cytotoxic T cells and with B cells. And then the suppressor cells help to turn that response off when you've cleared the infection, because you don't want to keep these really potentially dangerous hyperactive cytotoxic cells running around the body. Our B cells, they don't really directly interact with pathogens. except for their activation. Once they're activated, they're just going to be producing a ton of antibodies that are going to help other cells identify and destroy whatever antigen they're targeting. Those antibodies can also help to immobilize things like bacteria. So we find large clusters of lymphocytes in our lymph nodes. These are where we have a lot of afferent, which means signal in or fluid in. So a lot of afferent vessels delivering lymph to the central area of the node. And then you'll have one or two efferent vessels that leave out via the hilum in that lymph node. Now when you have these lymph nodes, you'll see within them these little clusters of lymphatic tissues. The purple is what you're seeing there. That are kind of interspersed between the stroma or the connective tissue component, or excuse me, the parenchyma or the connective tissue component of the lymph node. We do see clusters of these lymph nodes that occur near the body surface, which are the ones that we kind of feel swollen when we're sick, especially in the inguinal, axillary, and cervical regions. You will have clusters too in the abdominal pelvic cavity that are deep within the body. As far as those lymph nodes, their main function is to filter. So macrophages will help destroy microorganisms and cellular debris that have wound up in the lymph, and immune system activation, where we have our lymphocytes monitoring for antigens that can then develop a coordinated adaptive immune response. Structurally speaking, it's a little bit easier to take a look at this on the next slide where I have things labeled. So lymph nodes have two distinct regions, a cortex and a medulla. In the cortex you'll find lymphoid follicles which have germinal centers. Germinal means growing or developing. So this is where, like, when you encounter a disease for the first time and you need to clone a whole bunch of B and T lymphocytes, this is where that cloning process occurs. It's not where lymphocytes are going through development. That happens in the bone marrow and in the thymus, at least for T cells. But this is where, once they're out and functional, immunocompetent cells, when they're activated, they can then go through that clonal selection process, and they'll rapidly multiply once they've had a positive reaction with some kind of antigen. You will also find a subcapsular sinus surrounding this lymphoid follicle, and that's just kind of the space that that lymph percolates through. You'll have multiple of these follicles throughout the cortex, and then you get to the medulla, which will have other aggregates that are called medullary cords of lymphocytes, and then other sinuses that we call medullary sinuses. And then that will drain out through the efferent vessels. You can't tell efferent and afferent from a slide, so it's not something I would expect you to know on a histology picture, which I will throw in here in the next slide so we can take a look at that of one of our own micrographs from a lymph node. But it is normally encased in a little capsule. So these are encapsulated lymphatic tissue, which is different than MALT, mucosa-associated lymphatic tissue. And they have a pretty distinct appearance to them, and they're kind of small, so they should be relatively easy to identify as a lymph node. Now, these are actually from our class slides, and I pulled them off pretty recently. And so you're probably going to see these at this level, or maybe zoomed in one more step. So this is a 40x magnification, and I might use a 100x magnification for some of the smaller structures. But we can see everything we need to in here. And I wanted to show them both because the stains look a little different on each one. The one on the left is done with using a silver stain and some other things, so it looks a little more brownish almost. The one on the right is a standard H&E stain, which is what we see most of our tissues getting stained. In both of those, well, really on the right one, that light pink, that's what collagen stains. And so you can see that collagen matrix, the parenchyma, is just sort of running through the lymph node, helping to sort of septate it or divide it out into these kind of regions of the cortex. Now the one on the right is nice because it does show us where that blue arrow is. It does show us where those germinal centers are pretty clearly. The one on the left is really a good one, and that's most likely the lymph node I'll use for the quiz. The one on the left, the green arrow, shows us the medulla. The blue arrow is in the cortex, and it's specifically on a germinal center of one lymphoid follicle. So you can kind of get an idea what's going on there. The septa and capsule are way more visible in the image on the right, where you see them in bright pink. Now, lymphoid organs, such as your tonsils or the thymus or the spleen, come in a couple different varieties. The lymphoid organs that we're going to focus on, so tonsils are more closely related to lymph nodes. I mean, lymph nodes are organs too. There's just a whole bunch of them scattered around. These tend to be more consistent in their locations. Like your tonsils are in 99% of people always in the same place. You have the pharyngeal tonsil, the two palatine tonsils, and the two lingual tonsils. So those are all consistently there, and they don't have a capsule. So we call them unencapsulated lymphatic tissue, or frequently just anything in a mucous membrane like that we call a MALT, mucosa-associated lymphatic tissue. The thymus, which is in the thorax, this is big when you're a kid, and as you age, it gets smaller. It kind of involutes. The spleen is in the abdominal pelvic cavity, and then you have dispersed throughout the small intestines Peyer's patches. So the spleen is the largest of the lymphoid organs that we have. It's about the size of your fist, roughly the same size as the kidney and the heart. They're all kind of in that same fist-sized class. It is found beneath the 11th and 12th ribs. It is retroperitoneal and inferior to the diaphragm. And retroperitoneal means it's not actually in the peritoneal cavity itself. It's supplied by the splenic artery and then drained by the splenic vein, both which enter and exit at the hilus or hilum. In the spleen, you have a lot of blood filtration, and that's what we talked about it with in regard to chapter 19, I believe, with red blood cells and how they're formed and broken down. But it also is a site of lymphocyte proliferation. It basically is structured like a very large lymph node, and so when lymphocytes are in there and they detect pathogens, they'll go through that clonal selection too. It's a very good site because of its blood flow for immune surveillance and developing responses, and it does act as a kind of general filter for the blood. So we talked about the RBC function earlier chapters. It's just a reminder of that. It is also one of the main sites of fetal erythrocyte production. And then after birth, that transitions more to things like your red bone marrow. And that can be throughout the bones, but then as you age, it really just becomes the diaphyses, the epiphyses of our bones, the ends, and then some flat bones like the sternum where most of those RBCs come from. It also stores blood cells and platelets, which is kind of a cool thing. It's mostly storing them as a way to help you break them down. But when a cell is identified as too old and pulled out of circulation in the spleen, it will hang out there for a little bit so that if you do have a hemorrhage later on and you haven't broken down those old cells, it can contract and release those old RBCs back into circulation. Diving mammals like whales and sea lions and seals, they'll actually store a large volume of blood cells in there and then throughout the dive slowly release them, so they're releasing these oxygenated cells back into circulation. And it helps them stay under longer. Structurally, it looks like a lymph node too, although it doesn't have like germinal centers in the same way that we saw them in lymph nodes. It's an encapsulated organ. This is important because it has very leaky capillaries, and if that fluid could just leak out and swell up like a balloon, you'd bleed out pretty fast. That's also why splenic trauma tends to be pretty risky. There's a significant risk of internal hemorrhage. The structure of the spleen is it has these large vessels moving through it, and they'll often travel through connective tissue, what we'll call trabeculae, that help to kind of divide and support the matrix of the spleen. When you have this tissue moving through, you will have a lot of red blood cells that have been pulled out of circulation. Those are in an area that we call red pulp. And then you have areas where there's large aggregates of white blood cells, and we call that white pulp because of the cell type that we find there. Now, when you look at these histologically, be aware that white pulp will look darker because those cells have nuclei, and so they will just stain darker. Red pulp, like our red blood cells and blood slides, stains pretty light, and so it actually looks lighter. So the names and colors on a slide tend to be reversed. The thymus is a bilobed organ, so that means it has two lobes, one on each side, and it does secrete some hormones, thymosin and thymopoietin. Those are helpful in guiding the maturation of T lymphocytes and as they become immunocompetent. So this is when they go through positive and negative selection. When you're young, the thymus is big and well developed, and on the next slide we'll see one from a relatively young individual. it has, in that scenario, usually like these lobes with pretty clear, darker lymphatic tissue and then lighter medullas. So each lobe has a cortex and a medulla, unlike a lymph node where the whole thing was cortical with one middle medulla. As you age, you basically need to use it less, and so it involutes and becomes fibrotic and fatty tissue. By the time that you're in your, most of the cadavers I've seen in their 80s, 70s, the thymus is just, it looks like fascia, like just connective tissue at that point. And this, just to emphasize too, it doesn't really fight anything directly. This is just about making sure your T lymphocytes are ready to go. And it's a pretty tough process to get through. I think we're seeing something like a 95, 98% rate of filtration. So like only a couple percent of T lymphocytes that go there ever make it out alive. Here's a shot of the thymus. You can see again, it's very lobular. To me, it looks a lot like the pancreas. However, you don't see ducts and you don't see pancreatic islets in here. You'll run into these little structures called Hassel's corpuscles, which are little connective tissue or dormant cells. It's not quite clear what they are. But you'll run into those inside the medullas of these lobes, and they seem to accumulate with age, but their function's unknown. Our tonsils are the simplest of the lymphoid organs. They're unencapsulated. They're consistently in the same place, hence why we name them. But they're basically just this stuff called mucosa-associated lymphatic tissue. And it's just a place where lymphatic tissue gets really close to the surface of the skin, and you can monitor stuff that's going through chutes and tubes in your body, like your esophagus or your pharynx. The ones that we get in our pharynx are consistently in the same place, and so we name those. We have our palatine, lingual, and pharyngeal tonsils. The ones that tend to be more randomly dispersed, those just get called MALT, mucosa-associated lymphatic tissue, or GALT, gut-associated lymphatic tissue. You'll find those in any mucous membrane, like the ones that are in your respiratory system, urogenital system, and GI, gastrointestinal system. Now, those tonsils, again, they're not fully encapsulated. They have these crypts that can trap matter and then give your lymphocytes an early window into what might be going down the tubes in your body, or up, depending on where you're at. And those little like early warning signs help you mount a more effective immune response earlier. Now, you do get specific aggregates of lymphatic tissue called Peyer's patches that we find in the ileum, which is the last part of the small intestine. That is actually the easiest way to tell that you're in the ileum histologically, as you see these consistent patterns of lymphatic tissue showing up. But they're going to be really important for helping you kind of keep your gastrointestinal flora and fauna, the bacteria that are in there and single-celled organisms, in check. They're good for us, but like anything, it's the dose that makes the poison. Also, they need to be in the right location so our immune system helps keep them regulated. And then MALT, I think I've mentioned this acronym a few times, but it's used to describe these lymphatic tissues that are associated with mucosal membranes.